You know that incredibly frustrating scenario?
Oh, I think we all do.
Right. You get sick, you get prescribed a full course of antibiotics, you take them, and you finally, start feeling better.
The battle feels won.
Exactly. The battle feels won. But then, like, a few days later, the infection just roars right back.
Yeah, it's the worst.
And we're usually told to just chalk this up to, well, superbugs. Like, the standard explanation is that these bacteria are just mutating their DNA, right?
Yeah.
Evolving super fast to resist our drugs.
Right, that's the classic textbook answer.
But that always leaves a massive gap in the logic for me. I mean, how do they actually survive that initial devastating wave of medicine long enough to mutate in the first place?
Exactly. It takes time to mutate.
Right. And it turns out they aren't just, crossing their microscopic fingers. They're literally mailing each other survival packages.
It is wild.
It really is. So today we're doing a deep dive into some mind-blowing new research that completely flips our understanding of bacterial survival on its head. It's a 2026 science paper by Alice X, Wynn and her colleagues, and honestly, it is a masterclass in catching microbes in the act.
It really, really is. The source material we have today is it's dense, it is meticulously detailed, and frankly, it uncovers a survival mechanism that is both brilliant and, a little bit terrifying.
Yeah, a little scary for sure.
As we get into this, I've actually set the visual backdrop behind me to a dark, microscopic cityscape filled with these tiny, glowing, floating orbs. Because that imagery, those floating packages, is exactly what we are going to be exploring.
I love that visual.
We're looking at a system where bacteria act less like, lone wolves and more like an interconnected, highly communicative society.
And our mission for this deep dive is to completely unpack how that society functions from start to finish. We're going to hit every mechanism, every trigger, and all the crazy twists in this paper.
Nothing gets left out.
Nothing. So I want you, the listener, to imagine you have a population of bacteria. They are genetic twins, isogenic, completely identical in every way.
Right, same DNA.
But the exact moment they sense danger, they split. They just choose completely different career paths to ensure the survival of the collective. My first thought when reading this was, how do you even observe that?
Yeah, it sounds impossible to see.
Like, how do you catch microscopic twins sharing invisible secrets?
Well, that was the first major hurdle the researchers faced, right? And their solution was incredibly elegant. To prove that bacteria were actually sharing material with one another, they had to build a trap. A genetic trap.
Oh, the Cre-loxP reporter system.
Exactly. They used a Cre-loxP reporter in a strain of E. coli.
Okay, let's break that down for a second because the genetics can get super heavy here. I like to think of this trap like a locked door and a very specific key.
That's a great analogy.
You have two groups of bacteria in this experiment, right? A donor strain and recipient strain.
Right. So let's look at the donor first. The donor is an E. coli strain that is missing a specific gene. The GALK gene.
So no GALK.
Right. And because it lacks this gene, the donor cell absolutely cannot digest a type of sugar called galactose.
So galactose is just off the menu for them.
Completely off the menu. But the researchers engineered this donor to act as a factory for a very specific protein called Cre-recombinase.
Okay, so the donor has the key, the Cre protein, but it's starving because it can't eat the sugar.
Exactly.
And then you have the recipient. The recipient actually possesses the gene to eat the sugar, but it's broken.
Broken on purpose.
Right. The researchers literally flipped the gene upside down in the DNA sequence and put biological barcodes around it. These are the LOXP sites, right?
Yep. The LOXP sites act like a frame.
So the door is there, but it is firmly locked.
Right. So here is how the trap works. If the Cre protein somehow physically travels from the donor cell across the empty space and into the recipient cell, it acts as a set of molecular scissors.
So it cuts the DNA.
Exactly. It finds those barcode LOXP sites, snips the DNA, and flips the broken GALK gene right side up.
Wow.
Suddenly the lights come on, the machinery works, and the recipient cell can eat the galactose and grow. They become what researchers call gal-positive.
It is such a clever trap. I mean, if you see the recipient bacteria suddenly eating galactose, you have absolute proof that the key was mailed across the void and opened the lock.
Undeniable proof.
Now, when the researchers just mixed these two groups of bacteria together under normal, happy conditions, almost nothing happened. The transfer rate was incredibly tiny.
Yeah, something like 3.4 times 10 to the negative 7th. So roughly 3 in 10 million cells actually shared the key.
And here's where it gets really interesting. They didn't stop there. They introduced danger. They added antibiotics.
Specifically, they used sublethal doses. They calculated the minimum antibiotic concentration, or MAC.
That may seem right.
For a common antibiotic like ciprofloxacin, that was just 8.5 nanograms per milliliter.
Which is a tiny amount.
Oh, it's microscopic. Or they used carbinticillin at 5 micrograms per milliliter. It is just enough to stress the bacteria out, making them sound the biological alarms without instantly killing the whole population.
And when they added this mild stressor, the transfer rates absolutely exploded. They spiked by over 4,000-fold.
A massive, massive spike.
Which just blew my mind. But I do have to ask, and this is a bit of pushback, is this just some weird E. coli laboratory quirk?
That's a fair question.
Because we study E. coli so much since it's easy to manipulate, you know. But if this is just an E. coli party trick, maybe it's not a universal threat to human health.
The researchers anticipated that exact skepticism, actually. So first, they tested it in a stripped-down, bare-bones version of E. coli.
Oh, like a simplified one?
Yeah, the MDS42 minimal genome strain. It has all the excess baggage like leftover cryptic prophages and viral genes totally removed.
And did it still work?
The transfer still happened massively. But here is the truly concerning part. They then tested cross-species transfer.
Wait, really? Cross-species?
Yes. They used donors from a completely different genus of bacteria, Pseudomonas putida, and mixed them with E. coli recipients.
It still worked.
It worked flawlessly. The Pseudomonas bacteria successfully mailed the survival packages, triggering the genetic trap in the E. coli. They were communicating and sharing physical tools across species lines.
That is wild. Okay, so if we do a quick mini recap of our first subtopic here, Bullet 1 bacteria can definitely share physical material.
Right. And Bullet 2, that clever genetic trap, the Cre-loxP system, proved this sharing physically makes recipients capable of eating galactose.
And Bullet 3, which is the scary one, sublethal antibiotics, massively trigger this sharing network.
Massively.
So for everyone listening, I want to pose a question. Think about the last time you took a lower than prescribed dose of antibiotics. Maybe you missed a pill. Could you have been triggering this exact bacterial sharing network in your own body?
It's a very sobering thought. And it naturally leads us to the next massive question the paper tackles, right? What exactly is inside these packages?
Right. What is the cargo? In biology, information is usually shared as DNA or RNA or as a fully formed protein.
Yes.
Love a good spy movie analogy for this. If the donor is trying to help the recipient, are they sending the blueprint so the recipient can build the gadget later? That would be DNA.
Right.
Or are they sending an encrypted memo with the instructions? That's RNA. Or are they just mailing the fully built, ready-to-use gadget itself, which would be the protein?
To figure this out, the researchers used some brilliant biochemistry. They attached a chemical degradation tag to the Cre protein inside the donor cell.
The SSRA degradation tag.
Yes, the SSRA tag.
That's essentially like slapping a sticky note on the protein that says, hey, garbage disposal, eat me.
That is exactly what it is. The tag routes the protein straight to the cell's internal trash can, specifically the CLPP protease enzyme, which just shreds it.
So the protein gets destroyed before it can even be mailed.
Right. And when that happened, the transfer to the recipient stopped completely. But, and here's the genius part, when they genetically deleted the CLPP gene, removing the cell's trash can and saving the protein from destruction, the transfer resumed.
Oh, wow.
This unequivocally proved that the fully built gadget, the protein itself, is the cargo being mailed. It's not a blueprint. It's ready to use the second it arrives.
And the craziest part to me is that they don't even need to touch to pass the gadget along.
Contact independence.
Yeah. The researchers physically filtered out all the donor bacteria. They removed the cells completely, leaving nothing but the clear, cell-free liquid, the supernatant, that they had been floating in. And when they exposed the recipients to just that liquid, the lock still opened.
Because the protein isn't just floating naked in the water, it is packaged.
Right.
And to figure out the size and nature of this package, the researchers used a series of mechanical filters. The mysterious transferring activity passed right through a massive 1,000 kilodalton filter, but it got snagged in a smaller 100 kilodalton filter.
Okay, so kilodaltons are just a measure of mass for tiny molecules. So passing through the big one but getting caught in the small one told them the protein was part of a larger complex, much bigger than a single naked molecule.
Exactly.
And they figured out it was a little bubble of fat, right? They added a chemical detergent, something called NP-PEG, which basically dissolves lipids or fats. And suddenly, the transfer stopped working entirely.
The detergent popped the bubble.
Exactly. It popped the bubble, leaving the protein exposed and useless. It proved the protein was wrapped in a protective, armored lipid membrane, a microscopic life raft.
Precisely. We call them membrane vesicles, but to truly prove it, they had to isolate them. They used a technique called density gradient ultracentrifugation, or DGUC.
Right. They put the liquid in a tube and spun it at incredibly high speeds, like spinning a vial of blood to separate the plasma, or, how Italian dressing separates into oil and vinegar over time, but supercharged. Heavy stuff sinks, the light stuff floats.
That's a great way to picture it. And the magic transfer activity always settled in very specific lightweight layers of the tube, specifically fractions F2 and F3.
And what was the density there?
It was between 1.13 and 1.16 grams per milliliter, which perfectly matches the known buoyant density of bacterial membrane vesicle.
Okay, I want to use an image memory technique here. Listener, I want you to visualize this right now. Picture a glowing green orb safely tucked inside a tiny fat bubble, floating through the dark space between cells.
The glowing green orb is key because the researchers didn't just guess this existed, they physically photographed it.
Right.
They attached a glowing fluorescent tag called MVNUS to the Cre protein. Then they used advanced imaging, cryo-electron tomography, and single-channel nanoflow cytometry to take actual physical pictures of these glowing green vesicles.
It is stunning visual proof. Okay, let's do another mini recap. Bullet 1, the transfer material is definitely a fully formed, ready-to-use protein.
Bullet 2, these proteins are mailed in protective lipid bubbles called membrane vesicles.
And bullet 3, these vesicles don't require bacterial cell-to-cell contact to work. They just float across the void.
Exactly.
So my question for the audience here is this. If bacteria can package up functional, ready-to-use protein tools into armored lipid bubbles and float them across to other species, what other microscopic gadgets do you think they are sending to each other right now? Toxins, antidotes.
The possibilities are endless, and it radically changes how we view bacterial communities. But if we want to understand how this works, we have to look inside the factory. How does a single microscopic cell build and ship these armored packages?
Right, because bacteria don't have, like Amazon fulfillment centers. How are they actually manufacturing this? The researchers took those lightweight magic layers, F2 and F3, from the spinning tube and analyzed every single molecule inside them using mass spectrometry, right?
They did, and what they found was super weird.
Yeah, I thought this was fascinating.
Usually, when a bacterium sheds a bubble, it pinches it off from its outer skin, its outer membrane. But they didn't find standard outer membrane markers here. Proteins like OMP-A or OMP-C were mostly found in the denser, heavier fractions, not with our magic vesicles.
So if it's not the outer skin.
They found inner membrane and cytoplasmic materials, and one specific protein stood out above all the rest. PSPA.
Okay, so let's figure out what PSPA is doing inside these bubbles.
PSPA is the central player in a biological alarm system known as the phage shock protein response.
The thesis response.
Right. Under normal, calm conditions, PSPA just sits quietly in the cell, suppressing another protein, a transcription factor called PSPF, whose job is to turn on emergency genes. But when antibiotics hit the cell, they physically damage the inner membrane. This causes the cell's electrical battery, what we call the proton motive force, to start leaking.
Ah, so the antibiotic punches holes in the cell wall, the battery starts leaking acid, the alarms go off, and PSPA gets deployed to the front lines to do damage control on the inner membrane.
That's the setup. But PSPA isn't just a patch. It acts like a class of proteins called ESCRT3 proteins.
ESCRT3.
Right. In cellular biology, these proteins are the master architects of membrane remodeling. They can literally force a flat cellular membrane to bend, curve into a dome, and eventually pinch off into a sealed bubble.
Wow.
And the lipidomics analysis backed this up.
Lipidomics?
Yeah, studying the specific fats. The bubbles in F2 and F3 were enriched in a specific fat called phosphatidylglycerol, or PG, compared to another fat called PE. And it turns out PSPA preferentially binds to PG.
So PSPA is basically the factory foreman. When the antibiotics hit, it rushes to the inner membrane, gathers these specific PG fats, bends the wall, stuffs it full of survival proteins, and launches the life raft.
Yes.
And they proved this genetically, didn't they?
Mm-hmm.
When they deleted the whole PSP regulin, the genes that make all this happen, the vesicle transfer dropped by an astounding 8,000-fold. The whole mailing system just shut down.
It did. And they even did the reverse when they artificially forced the bacteria to overexpress a related protein, PSPC, which triggers the stress response. They started launching the rafts even without any antibiotics present.
They absolutely proved it. It is a highly regulated, intentional manufacturing process. But hold on, I'm stuck on something here. I really have to push back on the biology for a second.
Sure.
If the antibiotic is literally destroying the donor cell's membrane, how does the cell have the energy or even the time to neatly package up these perfectly sealed survival kits?
It's a great question.
Shouldn't it just be panicking? Like when I think of antibiotic stress, I always think of the SOS DNA damage response. The cell just throws everything at the wall to save its own life. Why isn't this just a chaotic death spasm?
That is a highly intuitive question, and it's exactly what the researchers wanted to untangle. Does the antibiotic trigger a general panic or a specific program? So they tested the SOS DNA damage response. While it's true that the antibiotic they use, ciprofloxacin, does trigger the SOS panic button, they found through genetic knockouts that the SOS response only plays a very minor role here.
Really?
Yeah. It is specifically the intermembrane stress, the leaking battery, that activates the HISPA response. That is the true driver of this horizontal protein transfer. The cell is dying, yes, but it uses its final energy reserves to execute a coordinated altruistic program.
That is staggering. Let's recap that. Bullet 1, the protein packages are actually intermembrane vesicles, not standard outer membrane ones.
Bullet 2, a specific protein, PSPA, acts as the factory machinery to bend the membrane and build them.
And bullet 3, antibiotic membrane stress, is the specific trigger that starts the assembly line.
Spot on.
So the question for you listening is, the donor is literally sacrificing pieces of its own internal organs, its inner membrane, to send these vesicles out as it dies. Why would a single-celled organism be so altruistic under fire? What is in it for them?
To understand the evolutionary logic there, we have to look at the other side of the equation. We know how the package is sent. But what makes a recipient open it?
Right. It takes two to tango. Okay, let's unpack this because this is where the story gets really incredible. The recipient actually does the exact opposite of the donor. To take up the life raft, the recipient cell has to actively suppress its own SPEEF stress response.
Yes. The researchers needed to figure out what was different about the cells that received the packages. To do that, they used single-cell RNA sequencing, or cRNA-seq.
But first, they had to actually catch them in the act, right?
Exactly. They built a new fluorescent reporter. They called it LoxP M Scarlet. The recipient cells glowed green normally, but the exact second they took in the protein package and the genetic lock was opened, they permanently switched to glowing red.
Oh, that's brilliant.
Yeah.
So you're looking at this microscopic sea of green cells getting hit with antibiotics. A bunch of them start dying and shedding packages. And suddenly, a few of the green cells pop red. And then you just grab those specific red cells, isolate them, and read their RNA to see what they're thinking.
Exactly. And when they looked at the RNA of these red cells, which they clustered together and called cluster 3, they found they were completely dormant.
Dormant, like asleep.
Yes. They had incredibly high levels of SRS, which is a 6S RNA that acts as a silencer during the stationary phase. But more specifically, they had massive amounts of a gene called HIPA.
HIPA. This is a fascinating protein. It's basically a toxin that the cell produces against itself, right?
That's exactly how you should think of it. It is an emergency brake. HIPA acts as a translation inhibitor. It induces a molecule called PPGPP.
Wait, PPGPP?
Yeah, it's an alarmon that physically shuts down the cell's ribosomes. And ribosomes are the biological factories that build proteins.
So no ribosomes, no proteins.
Right. If you shut them down, the cell stops growing, stops metabolizing, and enters a deep hibernation state called a persister state. The cluster 3 cells that took up the packages had almost completely downregulated their ribosomal genes.
So let me make sure I've got this straight. If a recipient cell wants to survive the antibiotic apocalypse, it physically hits the brakes on its own machinery and goes to sleep.
Yes.
And they proved HIPA was the absolute key here, right? Because when they artificially forced the recipient cells to produce extra HIPA, overexpressing it and forcing them to sleep, their uptake of the protein packages increased by 100-fold.
100-fold, and they even tested a mutant version, HIPA-7, which only targets one specific part of the machinery, GLTX, instead of the whole system.
And did that mutant work?
It didn't work at all. It requires a much broader metabolic shutdown to induce the right state to receive the packages.
That is just incredible. The genetically identical population splits into two entirely different states to ensure someone survives.
You know, I was trying to think of a mnemonic to remember this beautiful divergence, and it struck me.
Oh.
The donors are active, releasing the packages. The recipients are inactive persisters taking them up. Donors release inactive persisters, DRIP. It's a literal drip feed of survival. DRIP. Okay, that is actually a perfect way to visualize the division of labor here. I like that.
Right. Okay, mini recap time. Bullet 1 to receive a package, Bacteria must turn off their stress responses and go dormant.
Bullet 2, a toxin called HIPAA, acts as the master hibernation switch.
And bullet 3, the genetically identical population splits in two: active donors and dormant recipients.
Exactly.
So the question to ponder here is, to survive, the recipient shuts down its own ability to make tools and relies entirely on importing finished tools from its dying neighbors. In a crisis, is it better to work frantically to fix the damage or to board up the windows, go to sleep, and wait for help to arrive?
Which brings us to the ultimate survival test of the paper, right? Does this strategy actually work? Does this molecular mail actually save lives when the big guns come out?
The climax of the experiment, the lethal dose.
Yes.
So the researchers watched the cells and then hit them with a massive, devastating wave of antibiotics. A lethal dose of ciprofloxacin, 850 nanograms per milliliter.
Which is huge compared to the MAC.
Yeah, 100 times the minimum dose. And they left them in that toxic bath for 6 full hours. If they were going to die, this would absolutely do it.
And the results were undeniable. The dormant cells that had taken up the protein packages, the gal-positive ones that had turned red, they survived at a rate of over 40%.
40% doesn't sound like 100%, but...
But compared to the cells right next to them that didn't take up a package, it is a 28-fold higher survival rate.
28 times more likely to survive. And they proved this wasn't just a genetic mutation. The cells didn't magically evolve genetic resistance. They just had temporary tolerance, shielded by the packages.
Right, and they nailed the coffin shut on this theory with the preconditioning proof.
Oh, this was my favorite part.
They grew a batch of bacteria, stressed them out so they produced vesicles, and then extracted a liquid highly enriched with these specific vesicles, which they called P1.
Okay.
They took a totally fresh batch of bacteria and fed them this P1 liquid right before hitting them with the lethal antibiotic dose. And survival completely skyrocketed.
But conversely, if they took that liquid full of life rafts and added just 0.1% SDS detergent to it.
Which pops the bubbles.
Right, which destroys the vesicles before feeding them to the fresh cells. The survival superpower completely vanished. The bacteria died just like normal.
They even took mutant bacteria, the delta PS mutants, that were genetically incapable of building their own rafts. When those mutants were fed rafts from wild, healthy bacteria, they survived the lethal dose significantly better. The protective power is entirely contained within those floating packages.
Okay, I want to pause and just marvel at the magnitude of this. By stopping their own protein synthesis, these persister cells are basically playing dead to hide from the antibiotics.
Right, because antibiotics often target active machinery, so if the machinery is off, the drug passes right by.
But you can't survive forever with all your machines turned off. You still need some basic maintenance to stay alive. So wait. They stay alive by essentially drinking the fully formed metabolic enzymes, DNA repair factors, and ribosomes shed by the actively dying donors around them.
Yes, that is exactly the big picture dynamic here. In a crisis, energy is your most precious resource. In a world of information overload, this is the ultimate biological hack. The dormant cells outsource their survival needs.
Wow.
Building complex proteins from scratch takes a massive amount of energy. By taking in ready-made tools from the donors, they can weather the storm, repair minimal damage, and wait for the antibiotic to wash away, all without expending the energy to build those tools themselves.
It is the ultimate community survival strategy. Let's recap this final section. Bullet 1, taking up protein vesicles, directly causes a massive increase in survival against lethal antibiotics.
Bullet 2, adding extra vesicles to the environment artificially boosts survival even further.
And bullet 3, destroying the vesicles in the environment, removes this survival superpower entirely.
Completely wipes it out.
So, audience, if bacteria are using this protein-sharing network to survive our strongest drugs, how could we design a new medicine that hijacks their mail system instead of just attacking their walls?
It really opens up a whole new frontier of pharmacology. But it also changes how we view life at that scale. We've spent a century picturing bacteria as these solitary, selfish organisms, fighting a zero-sum game for resources. But this paper proves they are an interconnected, highly communicative society.
I am in awe of the complexity here. They aren't just lone wolves. They will willingly sacrifice themselves to send functional care packages to ensure the survival of their dormant neighbors during an antibiotic apocalypse.
It's incredible. And I want to leave everyone with one final thought to explore on your own. We discussed earlier how this horizontal protein transfer can happen across completely different bacterial species, right? We saw Pseudomonas putida mailing packages to E. coli.
Right. Cross-species.
Now, consider your own gut microbiome. You have trillions of bacteria inside you right now, representing hundreds of different species. Based on this research, it is highly likely that right now billions of different bacteria are passing microscopic life rafts back and forth. They are altering each other's biology, sharing traits, and saving each other in real time, all without ever swapping a single piece of permanent DNA. If they are constantly sharing their physical parts, what does that mean for how we define a single species of bacteria in the first place? Where does one end and another begin?
That is a phenomenal, provocative question. The boundaries are just completely blurred.
Absolutely blurred.
So the next time you look at a puddle of water, or, the next time you swallow an antibiotic pill expecting a clean binary victory, I want you to remember the murky, incredibly complex, invisible postal service fighting back inside. Let us know what you think of this, and until next time, keep diving deep.